Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. I'm absolutely delighted to introduce our speaker today, Dr. Adong Zhang, here from the University of Virginia, here in Charlottesville, Virginia. Adong earned her PhD from Purdue University in 1994 and is currently the William Wolf Faculty Fellow and Professor of Computer Science with joint appointments in the Department of Biomedical Engineering and the School of Data Science here at the University of Virginia. Her research interests focus on machine learning, data mining, bioinformatics, and health informatics. She has served as the editor-in-chief of the IEEE Transactions in Computational Biology and Bioinformatics Journal. She's the founding chair of the ACM Special Interest Group in Bioinformatics, Computational Biology, and Biomedical Informatics, where she served from 2011 to 2015. And she's currently the chair of its advisory board. She's a founding and steering chair of the ACM International Conference on Bioinformatics, Computational Biology, and Health Informatics, and served in that role from 2010 until fairly recently in 2019. Again, her research interests involve machine learning, data mining, bioinformatics, and health informatics. And specifically, Dr. Zhang develops data mining and deep learning, machine learning approaches to modeling and the analysis of structured and unstructured data with a variety of applications, especially as it pertains to biomedical applications, as is the theme of this year's Biomedical Data Science Innovation Lab. Dr. Zhang's presentation today is entitled Meta-Learning for Biological Knowledge Transfer. As always, we are streaming this lecture live and for recording via YouTube. And if you're watching on YouTube, thank you so much for joining us today. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are very much encouraged to submit any questions for Dr. Zhang via the chat feature in their Zoom sessions. I'll synthesize these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Adong. Thank you so much for doing this. We're really looking forward to hearing your presentation.
Thank you, Jack. Thank you for inviting me to be here, and I hope, I can be helpful for your lab. So anyway, so today I will focus on the machine learning part for bioinformatics and health informatics. So hope to talk about new things which may be helpful to the lab people. So first of all, I hope to mention that machine learning and deep learning has been popularly used in bioinformatics, and due to multi-omics data in different cases like cancer or other diseases. So we have plenty of data available. So people start to use machine learning, deep learning. To the main purpose of using that, or the main function this can do, is to integrate different modality data into a uniform representation, and you can do prediction. So with that, we start to think about, what are the challenges in this direction. So, so to apply the deep learning into this medical and biomedical domain, we do have a very challenging problem, which is called a big P, small n problem. So what we have is, we have an enormous amount of data, but each cohort, so we normally have a small number of samples, but each sample has a high dimensional features. So that's called a big P, small n problem. And moreover, in the image, with the images, you have a spatial relationship, but convolutional neural network normally can work well with that. But in the general omics data, you don't have that spatial relationship in the input data. So these are challenges to apply the deep learning approaches in biomedical field. So with those challenges, our goal is to design, novel models that can accurately predict based on the less training data. So that's our goal, how to predict based on the small amount of training data. So, it turned out that normally we can do this customized deep learning that can incorporate some domain knowledges into the more accurate prediction. So with that, I just hope to mention one paper we published in the top AI conference, top Triple AI. We published the paper which shows how to utilize both labeled and unlabeled data to train a deep learning model to make an accurate prediction. So in there, the central idea is how to make attention pooling layers inside the deep learning to make the model faster in converging to a prediction model. So that's one of the recent work we have done. But today, I hope to talk about the new things in machine learning and how this can be applied to our field, and we can design some projects in the lab coming in June. So the new trend here. So currently, if you have got into the deep learning, so you know, in order to train a deep learning model, you need a large amount of data to do this. But as I said, we don't have that much data in each cohort. So although we have a lot of data in general, but then we look at how humans learn things, even a kid does not learn things with a huge amount of data. So you can learn things very quickly. If you know how to ride a bike, you can ride a motorcycle, learn how to ride a motorcycle very fast. And if you see cat and bird, you can easily, just a couple of times, you can easily, quickly, tell them apart. So this is a human. So it turns out that you see human does not learn things based on a lot of samples, just a few samples. And in reality, in biomedical field, you do have the scenario, you have a big data, but it's a long tail shape. So small data are more common. So with that, the machine learning field has advanced a lot. So I hope to show what people are thinking right now. So basically, you have a few images, and then you just want to train, show you this is a bird, this is a dog, this is a cat, and then you can give in a dog and a bird. You can see this is a dog, this is a cat, right? And in our biomedical domain, if we have a few samples in different diseases, and then give you a new patient, you want to be able to detect whether this person is this disease or that disease. So with that, with limited training data, so what is our goal? Here, the interesting research has been going on is transfer learning. So to transfer knowledge from other domains to the domain you don't have much data. So transfer learning has been going on a lot. So the latest one is Meta-Renewal. Learning how to transfer. So that is one of the things we look at it. So what is Meta-Renewal? So Meta-Renewal as the new research topic in the machine learning domain, it's saying we want to mimic human learning by training a machine model on different learning tasks. But each task only has a few samples, okay? And then, so the machine can learn to quickly adapt to the environment with new tasks. So that's basically what we are training the machine. So to mimic how human learn. So to train such a model to quickly adapt with a few examples, that is a lot of challenge how you will do it, right? How you will do it. So a good, there are different, I will skip this one, how we will to next one. So how we do it. So basically, I want to use this example I borrowed from a blog. You know, it's a good example to show how this works. And here, as the explanation, rather than going through the formal, you know, processes. So basically, you have, you know, if you are familiar with machine learning models, you train the model using a lot of data. Now, I don't give you, you know, a lot of data to train model. I give you a lot of tasks. So you can have 1,000 tasks, but each task has a training support set and a query set. So for example, training task 1 has this is called n equal to 3, means n way, which is number of classes. So you have 3 classes. K2 means 2 samples. So each class has 2 samples. n way 2 shot. So this is what when people saying n way. So each task, I only have 2 samples in each classes. I have 3 classes. And the query set, this has a 1 from each class. Okay, so then the training task 2 can have a totally different samples from the task 1. So for example, you can see here are the dogs, dolphin, and lions. So these are very different from the task 1. So you have thousands of such tasks to train your model. And then you have the test task. Similarly, you have a support set and you have query set. Okay, so with that, so you train the, so each time you use a task and then you use the query. So the machine can look at the support set and the query set to quickly adapt to detect the query set. And then you use the second task to quickly adapt. So in the end, the model is not trained for any specific task. It's trained in general to detect, to adapt to the new environment. So when you train your model can do this, then when giving you a query task with only a few samples, then you can quickly adapt your model to detect when given query. Okay, so that's generally what a meta-learning means. So it's trying to mimic how human learn. So with that, I hope to mention there are different versions of meta-learning, you know, in the coming from the machine learning community. So the memo is the most famous one from Berkeley, and then you have a few shot learning methods. These are all different methods. And so I will use the memo as an example to show how it works. So basically, as I said, you train the model to quickly adapt. So what does that mean in terms of the parameters in the model specifically? So you can see here, if this theta is what we want to train, right? And then using those support tasks, we quickly adapt to each environment. This small delta L's, you can quickly. So with this adaptation, quickly you train this theta. So eventually, after the training, you got a theta, which is your meta-renewal. After meta-renewal, the model, which is supposed to be able to quickly adapt to new environment. So then after that, when you are given new tasks, so with just a few samples, this theta can quickly adapt to theta 1, star, theta 2, star, theta 3 star in different environments. So if you're familiar with the transfer learning, this is a similar concept. That means we will learn a good theta in the model, which can easily adapt or fast adapt to different environment. Okay, so hope that it's clear on what the basic idea of what meta-learning is. So recently, so meta-learning here has been applied to computer vision a lot. So very popular approach. So, but recently, in the past 2 or 3 years, it has started to be used in the biomedical domain. So here I reference 2 papers. They show the meta-learning outperforms regular transfer learning in their paper and with data. The second one is a significant work from Stanford. So basically, they are showing the meta-learning. Using meta-learning, you can transfer the knowledge from different cell types. So you can adapt to new cell types with just a few samples. Okay, so I would specifically mention this paper because you know, you're doing neural-related research in this lab. So this paper applied meta-learning to the Alzheimer disease. So basically, this paper, so I'm using their slides, so it seems automatically moving. So this slide, they actually use the amnesia and dementia and Parkinson's samples as their support domain. And then they use the MCI, which is very close to Alzheimer, as the support. And then, so to train the model using these 3 samples, which are similar, but you know, in the same category, but a different kind of different disease, to train the model to extract the common the parameter which can quickly adapt, then eventually used for Alzheimer. So that's what they designed. So in their back to the slide. So in this, you know, they have a slightly different design because they use the MCI, which is more closer to the Alzheimer as a test domain to first train the model and then adapt to Alzheimer. So in their paper, so they used so here's the data they used from Oregon Health. And so you can see from the data set, they have dementia and amnesia. So these are all not a lot of data, so case and control. So each of them, you know, you won't be able to, you know, train a deep learning model with each individual cohort. But with the way designed here, using the source domain to train them to train the meta-learning model, they are able to use this amnesia and dementia data to train the common parameters which you can quickly adapt to the Alzheimer disease. Okay, so this is what they experimented. So with their result, reported from the paper. So they compared with a lot of supervised learning models, so including all this logistical regression, KNN, random forest. So these are all popular approach people use. And then they also use some other basic meta-learning approach. And they showed, so their meta-CNN and the meta-LSTM. So you added the meta-learning process into the model on top of CNN and LSTM will do much better in the prediction of diseases. So they show the result like that. So this work showed that the meta-learning is effective in the biomedical applications. So with that, we also published a paper, to show how meta-learning works for Tcga. So if you are familiar with Tcga, this cancer genomic atlas, so it has over 20,000 primary cases, cancer samples, both control and disease cases, and they have plenty of features. So they have genomic, they have epigenomic, transcriptomic, all proteomic, they have all these features. So the features are enormous, it's a high dimension. But if you look at although they have 20,000 samples, but if you look at each category of the cancer, so these are different cancers in there. So they have 33 different types of cancer. But if you look at each cancer, the samples are quite small. The largest is just over 1,000. So it is not practical to train any of these cancers using the deep learning model because the samples are too small, right? So how do we utilize this in prediction models? So we look at it carefully, and so you have all these different data types and features, and then we apply. So our goal is to perform cancer prediction. So here, so we, in order to do that, we utilize the label the samples from different cancer set. And so, for example, here you have the BRCA cancer, you have this GBM cancer, and this different, so these are the full name of the cancer. And then applying the same meta-learning process, so we train, so we form a different set of tasks with each different kind of cancer. We can form small tasks, and then we can train the meta-learning model and do meta test, and then form a model which can quickly adapt to the new cancers. So that's the idea. So in a little bit more detail, I'm not going to show the formulas, but I will show this picture to illustrate the idea. So basically, in the episode is the term they use in the machine learning community. So in each of the inner loop, you train the model using tasks. So these tasks are from different cancers. So each cancer, you probably use just a few samples, and then you can train it to quickly adapt to the query, to predict the query. And then in the outer loop, you use the different tasks to test, and then it can. So this trained theta will adapt quickly with the new testing cases, And then with that, you got so you gradually train a good theta, which is you can use to adapt to any new cancers. Okay, so this is what is applied to the Tcga using the Meta-Renewal framework. With that, so the training setup in our environment, so we have all this different data. So we use the 31 cancers from the, so there are two, I think, too small and or not, good cases to be used. So we randomly select 21 cancers, and then we use the validation, randomly select 5 cancers, and then the testing case, we randomly select 5 cancers. And so with that, the result, we tested with different features. So first, using just the proteomic features as the features of samples. And then we have this many patients from 31 cancers, and 3-way, 5-way means we use 3 classes to train the model, and it means 3 cancers each time. And 1 shot means each time you have 3 classes, 1 sample in the class. And 3 shot means 3 classes, 3 samples, or 5 classes, 1 sample, and 5 samples. Okay, so with different scenario, we see how it works. So we compared with different meta-renewal method and also KNN random forest. And so we use the, so mammal-based perform the best in the scenario. So these two, the matching network and prototypical network also are meta-renewal methods, but it looks like a mammal-based it works the best. So here you can also see when you have more samples in each class, it does perform better. So this one has 5 samples in each class, and you train the model, it does perform better. So that makes sense, right? You have more samples in each task, you can train model better. So these are remarkable, result from using the meta-renewal to do the prediction. So we also use the, more different, set of features. For example, this one, we also including the RNA expression data. So we have more features, and then did this. So with these features, more features included, it looks it's performing better, and even reaching 98% accuracy in here. So that feature. And we also use all features, including everything in Tcga, the available features. Then actually the performance drop a little bit, because so you can see some of the features are not useful or duplicate or noisy. So we saw those noises. So this is what is showing with that. So it is quite encouraging to use meta-renewal to extract common good transferring capabilities to transfer to new cancers in this way. So we've shown that, other people also shown that. So with that, the current research, what current research we are interested in. So we want to not only just purely use the meta-learning approach, the machine learning community design, we also want to integrate knowledge from the biomedical domains. So the issue is how you will integrate the domain knowledge into this process. And so one thing we can look at is domain knowledge formed by graphs. So for example, in image, you can have this hierarchical graph, mammal to dog have different dog, cat have different type. So from patient data, just look at the nervous system, you will have a different kind of disorder or degenerative nervous system and Alzheimer. So this belongs to different category. So you do have such an ontology as knowledge you can use, and you can incorporate that into the systems. So, so with that, we look at the mimic data. So mimic data is clinical data. So these are including mostly the ICD-9 diagnosis code and lab testing result. So mimic data has a lot of disease and a lot of features too to be used. So with that, the reason we use this is because mimic data, they coded based on the disease. So the code actually indicate what are the categories. So for example, nerve, they have 3 numbers and go to 4 number. This is indicating different diseases. So in the hierarchy. So we can utilize this in the Meta-Renewal system and to narrow down our so I will not go to detail. This is a KDD paper in KDD 20 paper we published. So I will just show some result. So with those knowledge integrated into the process, we can show that. So this is Tcga net is the one we included into that learning model. So we can narrow to the different ontology relationship. So to utilize those relationships, you can see our approach perform better than mammal. So previously we showed the mammal-based approach outperform this approaches matching network prototype. So you can see they basically mammal works better than this in the example, but ours works much better because we utilize the domain knowledge in the process. So here subgraph sampling means we sample the data based on the subgraph definition. So if you only sample based on, so here and random sampling case. So here is what it means. If I sample just tasks based on the subgraphs, so then they are more similar. So it's more difficult to distinguish between them. So if you randomly sample among the graphs, then they are more far away. It's easier to distinguish. So in that sense, why when you use subgraph sampling, we get lower accuracy than random sampling. So, but we want to show, with different strategies and with the graph integrated, how much it affects the results. So this is what we get with that. Okay, so if you want to learn more details, you can look at the paper. This is a KDD 2020 paper. So with that, let me see. We also so this is the mimic data. We have this predefined ontology, but I mentioned the cancer data, and we want to apply the cancer, some kind of ontology on the cancer data too. So this, I just want to mention, the cancer also has maybe not a predefined ontology, but they have, based on the data set, you can build hierarchy with the data. So we did not do that, but I hope to point out, if you are interested, you can look into this, how to build ontology based on the data in the TCGA for this purpose, because there is no predefined relationship between the cancers in general. So you have to build based on specific data. So this is a potential interesting research people can look into. Lastly, before I finish my talk, I also to mention a potential research, for the lab people can think about, which may be interesting to you, because this can be applied to both cancer and Alzheimer or other diseases. So that is how to apply the Meta-Renewal approach for survival analysis. So basically, the survival analysis have some unique features. So the deep learning may be good to apply. So for example, in the TCGA pan-cancer dataset, or in the ADNI and ADSP disease dataset, so you have a lot of features, and you have a lot of samples, and you also have this survival data indicated when the sample died, and but the challenging issue is you have probably 78% are censored. So censored means the subject leaves before event occurs. So you don't know when they died, so or when they had the Alzheimer disease. They left early. So those in the survival analysis domain is called censored data. So survival analysis has been going on for a long time. So there are many survival models. Cox is the most famous model. So the issue is how to integrate those models into the Meta-Renewal process. So the basic concept, you have the censored and uncensored patient. So if you only use the uncensored patients, which when the event happened, then that's more accurate. But the problem is, as I said, most of the data sets don't have that information. So how do you deal with such a censored data? You probably don't want to just get rid of it, because then you lose a lot of information. So here we actually proposed to use deep learning to directly learn a survival distribution from data, not using the Cox model, because you can learn the distribution just utilizing the best time they left. So when they left and a sample left earlier than another sample, you can consider that has a different time of event may happen. So the later one will have a later sample. So using those relationships, we can learn using deep learning model for the censored data, make a prediction of the distribution. So that's what we currently working on. So basically, so since patients live up to the censored time, so we just in our deep learning model, we just use the log likelihood of their survival probability before censoring time as the log likelihood. So we are maximizing what we know based on the data. Then we use ranking to rank the relative risk of patients. So that's how we deal with censored samples. In fact, there are other papers they recently show this can perform better than the Cox model utilizing the censored data in the survival analysis. So this is what we do. So in the model, I will not go details about the model, but we will. So basically in the formula, we will indicate which are the censored, which are not censored, and then we can build an optimization function to learn the distribution based on the data. So that's what we have. How we handle the survival model part. So with that, so the matter survival learning. So in order to incorporating that into the matter learning, so we have as I mentioned, the matter learning and the base learning models are introduced in a way they can train and to be able to adapt to the new environment. So, but in order to integrate the survival analysis into the matter learning, so in the matter survival, the survival model is designed, so survival model is as a base learner. So you learn the base survival analysis based on what I just mentioned. So so you will make predictions for different patients with different cancers, for example, or Alzheimer diseases. And then using the same principle, the survival model will fast adapt to the prediction context. So it's a similar principle. The only difference is that you train that survival model inside of the matter learning. So so then the prediction made by the survivor will continue on a specific disease under the matter learning process. So so that's the general idea. So now here what we are working on here is we try to use knowledge more into the matter survival model. So we have so now more customized here, not very general, because in terms of the biomedical, so we build a knowledge repository, which indicating so the related samples. Okay, so the samples in a customized way from the repository, so to construct some context. So in order, so that if we can construct such a context for survival prediction, then we can make a better prediction model. So specifically, two kind of the sample knowledge or context we build. The first one is difficulty awareness. So this we can recognize, prediction difficulty of a given patient sample related to other samples in the repository. So so we have the set of patients, then we can assign attentions. So if you have learned this attention model in the NLP modeling, so we assign attentions to each of the samples. So we build a sample set which, to realize the context of difficulty in the samples. And in addition to that, we also indicating the heterogeneity awareness, which identify how a given sample is different from other samples in the repository. So then place the survival model at a proper position in its parameter space. So these are the current contexts we consider the model. So to be a little bit specific, this is a general framework we have been building in our meta survival analysis with knowledge. So so here you can see here, I strongly believe knowledge from the biomedical domain can play a very big role in the machine learning process. So, and how much this can generalize to different, different domains of diseases, that's a different issue. But the general framework should be able to apply to different domains. So basically, on the left of this figure, you can see we can track the context of samples, and with the graph of knowledge in the domain, and then we can integrate this into our training of the meta-learning process, and so with difficulty and heterogeneity. Then also we incorporate we can incorporate the survival model and into this process. So that's currently what we are building for the survival analysis model. So we have some results, but I do not have the tables to report, a complete table to report here. But I strongly believe incorporating the domain knowledge is very effective to build a better meta-learning model for knowledge transfer. Okay, so that's mostly, I hope here they encourage some of the, some of interest into this research for the lab. So now I'm going to finish up with my slide here. So basically, in a conclusion, so we demonstrated the ability of meta-learning, memo and other parts. So how to predict cancer type and survival time. So that's so then with this we can build a model that can classify rare cancers. So that's the main point, to classify the rare cancers with new subtypes. You obtain just a few samples, you can quickly adapt to the new samples. But I should point out that there's still a long way to go. There are many interesting problems in health and bioinformatics for which so the new machine learning methods can help. I do think that the new machine learning methods can help, but it does need both, the very close collaboration between the computer scientists and the domain experts, so that we can incorporating domain knowledge into the prediction models. So that's the whole point I hope to make here, since the lab has both data scientists and biomedical domain people. So I think working closely between the two sides, we can generate good ideas and results. So with that, hope to mention some of acknowledgement. So basically the NSF support, and also my students who contributed to this presentation. So I basically finished based on the budgeted time. Hope I will be able to answer some questions.
Hey, Dong, thank you so much. That was wonderful. Really great synopsis of this kind of meta learning applications, and you covered a lot of ground. Thank you so much. One of the things that I was kind of wondering as you were presenting is when you kind of know beforehand what your training samples happen to be, like where you have the different types of cancers, or for example, your early example of looking at the faces of animals or something and trying to identify kind of what's the minimum amount you need in order to kind of get the meta learning thing started. Nevertheless, you kind of have to kind of specify a priori. At the same time, that's kind of a static, that's a static data set in a sense. And then you hope to be able to apply it later on to, new exemplars. I guess where I'm going with this question is, this almost seems like it would have a lot of applicability in streaming information. As you're getting information, it's kind of like flowing in to a system and being able to, in a very quick amount of time, begin the bootstrapping the meta learning process, and then being able to perform classifications as data is streaming in. Do you have any ideas about how you might apply that? And there's a number of like biomedical or neuroscientific data types that might be like that might be very appropriate for a modeling system like this. Does that make sense?
Yeah, but that's an interesting question. So basically, you're thinking you have a streaming data. So, but are you assuming the streaming data will be new type of data? Or it's the same data?
It would be, well, for example, it might be electroencephalographic data. So it's multiple channels of electrical activity coming, from someone's brain. Could one build a meta learning system that starts reading that in and starts using it as it's streaming in? The person's performing, behaving or thinking, or there's mental activity going on. And the system could use that information to perform meta learning about generalized things. But then when asked to do specific things, the model might adapt and learn from it itself.
Yeah, that's a very interesting, thinking, because I haven't seen people doing that, but, in principle, you are streaming, you have different type of, data coming, right? So you can form tasks, in terms of tasks, different tasks, and to extract the most common features among these tasks, right? And then you can quickly, jump into the new data coming, and you can make a prediction. That's
what I'm kind of wondering is you could have the system just start not knowing anything and just start reading the stream. And as there's different tasks or something are happening, it might be able to learn the general principles, and then you could you could build up a knowledge base of how that stream should be coming and interpreted, and then you could throw curveballs at it, meaning you could change the task slightly and see how quickly it adapts to the new task.
Yeah, I think it can work, but this need to experiment.
Of course.
I think in principle it should, it's a potential application.
Yeah, just thinking about that as a possibility. Also, what you mentioned, it can also be kind of used to look at images. You gave an example, of course, of animals. I'm curious if you've attempted to apply this to any, say, microscopy data coming from microscopes to do, look at different cell types or something.
Yeah, we did not do that yet, but certainly this is one kind of application you can do. Yeah.
I mean, there's plenty of that data around, so it would be kind of interesting to see it applied.
Yeah.
And another thing would be like in human neuroimaging, where you have time series data, for example, or you have different subjects, different patient groups, for example, and being able to use this to try to classify some of the neuroimaging data.
Yeah, that's actually, so in the computer vision domain, originally this Meta-Renewal was proposed, was used for those common images or different images. So I think it will be very likely to be effective in those, images you just mentioned. Yeah.
Hey, Dong, what do you see as being an important element for biomedical scientists where data scientists can play an important role?
I guess, yeah, that's a very important question. So, in fact, I just taught a class for biomedical department. They created this new course called Data Mining and Machine Learning for Biomedical Engineers. So, yeah, I just finished that course, and I believe there is a gap, between the data scientists and the biomedical researchers, because I feel the tools biomedical scientists use are still very basic tools, and so mostly statistical oriented tools. So I believe there is a gap between how much you can use, from the machine learning data science side to the biomedical research. So that really need both sides work very closely in the problem solving, I think.
Yeah, I appreciated your comment there in your conclusions. You have it up on the screen now. That close collaboration is really important between computer scientists, data scientists, people doing analysis, so that these machine learning methods can be kind of incorporated, that those prediction models actually mean something and the predictions are valuable. But you're not going to get that unless there's close collaboration and team science kind of approach. Would you agree?
Yeah.
Fantastic. Well, with that, Adong Zhang from here at the University of Virginia, thank you so much for sharing your work with us today. That was really fantastic. For everyone who joined us, thank you so much for doing so. Everybody have a wonderful weekend, and we'll see you next week for our final in this series of Biomedical Data Science Innovation Lab Seminar Series. Take care, everybody. Bye-bye.